Hello, students. Welcome to Missology. I'm uploading the third set of questions for brilliant scores in biology. All you got to do is take a pen and paper and start writing the answers to these questions in their serial order. You should not be devoting more than 40 minutes for these objective types of answers to be written. You got to follow the serial order while attempting the questions, and moreover, there'll be certain formats in which you got to write the matching and the odd one out. So I will come up later after you would have finished writing the answers, providing you with the perfect answers so that you can tally and find out your scores. This will give you a very proper check of your progress as well, so that you can further revise depending upon the performance in the tests given to you. Hello, I'm back. You may match your answers with me. The answer to question number 1, it is thick cuticle, which is the adaptation in the banyan leaves to prevent excessive transpiration. Answer B is the intensity of light, which is the factor that leads to solarization in plants when it is excessive. Subpart 3, it is plasmolysis, that is the process which helps in preserving pickles for a long time. Answer D is NADPH, nicotine amide adenine dinucleotide hydrogen phosphate, that is the reducing factor in photosynthesis. And finally, subpart E is stroma of chloroplast, which is the site of biosynthetic phase. Question number 2 is match the words in column A with those in column B. Here, let me remind you again that you got to make that table and just shuffle the terms under column B so that the befitting word comes against the terms of column A. So IAA, that is indole acetic acid, should be matched with cell elongation. Then ABA, abscisic acid, should be matched with stomatal closure. The third one, cytokinin, should be matched with cell division. The fourth one, ethylene, to be matched with tuber sprouting. And finally, the gibberellins, it should be matched with internodal elongation. We move on to question number 3. Here you got to give the exact location. So in this exact location, we have to write the location of the palisade cells. These palisade cells are present below the upper epidermis of a leaf. The next one is the pulvinus. Pulvinus is located at the base of the petioles of a leaf. The third one is the guard cells. These guard cells are found surrounding a stoma, which is mostly present on the upper and the lower surfaces of the leaf. Then you go to hydatodes. For the hydatodes, they are present on the margins of a leaf at the extremities of the veins. The last is stroma. Stroma is located in a chloroplast, which is the ground matrix of a chloroplast. Then we move on to question number 4. So in question number 4, it is all about filling up the blanks. So you have 10 different blanks and you got to supply the word. Here you may just supply the word, putting against them the different A, B, C, whichever is mentioned. So 4A is kill the cells. 4B is methylated spirit or methylated alcohol. Part C is to remove chlorophyll, or you may write removal of chlorophyll. Part D is soften, SOFTEN. Part E is iodine solution. Then part F is variegated leaf. Part G is green. H is bluish black. I is xanthophyll. J is light brown. So I'm repeating once again for all the terms for the fill in the blanks. Kill the cells, methylated spirit or alcohol, then removal of chloroplast, then soften, then iodine, then variegated green, then bluish black, then xanthophyll, and finally light brown. So we move on to question number 5. This is about giving the differences with the point which is mentioned in the bracket. So here, when we come about the differences, the first is to difference between the stomata and the lenticels with respect to its location. So for stomata, you got to write abundant on the lower surface of a leaf. And for the lenticels, it is on the woody stems. Then it comes to the turgor pressure and the wall pressure. For this turgor pressure and wall pressure, when you got to mention its direction, so the cell content to the cell wall. Turgor pressure is exerted from cell content to the cell wall, whereas the wall pressure is exerted from the cell wall towards the center of the cell. Then it comes to the third pair in the difference. This is about the process osmosis and active transport with respect to the type of membrane. For osmosis, we require a semi-permeable membrane, but that semi-permeable membrane can be either living or non-living. But when it has to perform active transport, it requires a selectively permeable membrane, which must be a living membrane. Then subpart D, it is about exosmosis and endosmosis. Here also you have to mention the movement of the water molecules. So in exosmosis, water molecules, they move from a cell to the exterior environment. And in endosmosis, the water molecules move from the external environment inside a cell. Then finally, the last pair is heliotropism and thigmotropism. You got to differentiate based on the stimulus. So for heliotropism, sunlight is the stimulus, whereas for thigmotropism, it is the touch receptor, or the touch is the stimulus. We move on to question number 6. Here, this is based on a diagrammatic question. So, here in this diagrammatic question, let us see what are the following questions. The first is you got to name the aim of the experiment. So here the aim of the experiment is to show that oxygen is produced during photosynthesis. And since the process is photosynthesis, you got to define it. So photosynthesis is the manufacture of glucose by plants making use of raw materials from the environment like carbon dioxide and water with the help of sunlight and chlorophyll. Next is to name 2 plants that can be used in the setup which is shown. So it has to be aquatic plants. You may give the examples of Hydrilla, Vallisneria, Elodia, any 2. The third one is, what is the gas which is evolved and what is its source? So the gas which is shown is oxygen and its source is water. So splitting of water produces oxygen in photosynthesis. So the source is water. The next question is, what will happen if we increase, that is the bubbling will increase with a pinch of sodium carbonate added to the beaker, because this will increase the percentage of carbon dioxide. But yes, if the percentage of carbon dioxide goes beyond 0.03, it may even lead to the fall in the rate of photosynthesis. Here the last one is, what happens if this arrangement is covered by a black cloth or it is removed away and kept in a dark room? So photosynthesis will completely stop and there will be no evolution of oxygen at all. Then we move on to question number 7. Here, question number 7 where you have to find the odd term and then give the category for the rest. Here again, a table is required to be drawn. On the left, you got to mention the odd term, and towards the right, you got to mention the category for the rest. So, in the first case, the odd term is crista, and the rest of them are the structures in the chloroplast. For the second one, it is polymerization, which is the odd term, and the rest category, they are the steps in light reaction. For C, hydrogen is the odd one. For the category, these are the products of photosynthesis. Subpart D, odd term is cotton, and the rest of them are types of variegated leaves. And the last one here, where you have to differentiate again, you have to find the odd term. The odd one is indeed grasshopper, and the rest of them are autotrophs. You are given wheat, corn, grass, grasshopper. So grasshopper is a herbivore, so you don't have to write that. You just have to take out grasshopper, and for the rest you have to write autotrophs. Now let's move on to question number 8. Here you again have a diagrammatic question for which you have to supply the answers. So you got to predict the nature of the solution named X. This is the solution which is inside the cell wall, but it is outside the cell membrane. This has to be hypertonic solution. Do you have a justification? Because when osmosis is happening, it is only the cell membrane that chooses if water will come out of the cell or not. So if outside the cell membrane it is not laden with hypertonic solution, the cell will not undergo plasmolysis. Now, subpart B, you have to label the parts number 1 and 2. 1 is cell wall and 2 is cell membrane. The third part of this question is, what is the relation between the solution named Y with that of the solution X? Both the solutions are of the same concentration. That is, the nature of both of them will be identical, because the solution from the external environment enters through the freely permeable cell wall and comes close to the cell membrane, which leads to plasmolysis. The last is, number D of this is, you got to define the process which is shown. So the process itself is plasmolysis, and you got to write it is the process where the cell loses water to the exterior environment with the detachment of the cell membrane from the cell wall. And finally, you are supposed to mention one use of the process that is shown. So, plasmolysis can be used in the preservation of pickles, or even in the preservation of other food items. Thank you for watching. Please do like, share, and subscribe to my channel.